afternoon, and thank you for your attention. My name is Uretsky. I'm a neuro-ophthalmologist, and I'm the Director of Clinical Development at Genesight Biologics, which is based in Paris. And I want to start by thanking the organizers of the meeting and President San Antonio for inviting us here, along with Professor Corelli for inviting us here. It's always wonderful to see Professor Corelli and his team. And I think most of all, I want to thank the patients, many of which I met today. I think patients, especially in rare diseases, give us a lot of energy to get up out of the bed every morning and go and do what we have to do to try to find treatments and cures for their disease. And I think, especially in rare diseases and orphan drug diseases, they're so critical to develop in drug development, as has been said here today. So with that, I'd like to tell you about Genesight a little bit and what we're doing. Our goal is to develop therapies and treatments for mitochondrial diseases, neurodegenerative diseases, and diseases both aging and genetic of the eye and the retina. And our two main products are GS10 and GS30. I think GS10 is a little bit more interesting to this crowd. It's an AAV-based therapy. We've heard a lot about AAV therapies today, and that's because it works. It's a gene therapy, and in general, at Genesight, as the name would imply, we focus on gene therapies. And we're at the end of, nearing the end of phase 1/2A, and going towards phase 3 very quickly. Our other platform, which is very interesting, is an optogenetic platform, which also employs AAV-based technologies for degenerative diseases of the retina, such as retinitis pigmentosa and geographic atrophy. In that case, we're in the regulatory preclinical stages. It's really great because we have a fantastic network around the globe of very motivated and intelligent people that we work with. This is both from the preclinical stage and the basic science stage all the way through the clinical stage. In fact, we work with a fantastic group here, Professor Corelli's group with Piero Barboni and their group, but also in Germany with Thomas Klopstock, in UK, I know there's some UK folks here that I've met here, with Patrick Mann, in Paris where we are with Professor Sayel and his team, and also in the States with Alfredo Sadoun, whose name's been mentioned many times, and Nancy Newman, the president of Nanos, and also with Mark Moster, who's at Wills Eye Institute, which is where I trained in neuro-ophthalmology. So I know there's a mixed crowd here, and for some of you this might be basic, I apologize, but it was really geared towards the patient. I know the scientists and the physicians in the room think of the retina as an extension of the brain, and it is. Because if we look over here at this gross schematic, we have the sensing cells, the photoreceptors that are absorbing the light and passing this electrical information along through the interneurons, which are already doing a lot of processing of the information. And they hand it off to the retinal ganglion cells, whose axon form the optic nerve and take all the information back to the brain. So, unfortunately, in LHON, we have the mitochondrial dysfunction leading to the death of retinal ganglion cells and loss of central vision. And at the other end of the retina, the outer retina, we have degeneration of the photoreceptors in diseases such as RP and macular degeneration. Our approach is to use gene therapy, and again, this is more a patient-geared slide to help them understand what that is. And we heard a lot about AAV-based technologies, again, because they work. And I like to explain to patients that the AAV vector is a virus that's like the cars. It's the vehicle. It's modified so that it's not infectious. And the gene, the normal gene that we want to replace, is the passenger. And the vector is going to drive the gene, the passenger, to the cell where we want it to go. And obviously we're all using different AAV vectors based on what cells we want to target. The eye is a a little bit easier than most places, particularly the brain, because there's ways to easily inject medications into the eye using standard techniques that are employed in diseases such as macular degeneration. So there's been thousands, probably millions of patients who underwent injections with this technique. So these two platforms are very exciting because we have the ability to leverage them, not just for these initial diseases that we're looking at, LHON and RP and AMD, but we can then expand them and apply them to other mitochondrial diseases, to other neurodegenerative diseases, and to other degenerative diseases of the retina. So in GS30, the platform is based on optogenetics, and the goal is to replace the light sensing ability of the retina with the AAV as a vector and a stimulation device. And in GS10, again, a little bit more interesting to this audience, we have a proprietary mitochondrial targeting sequence which allows us to now take the gene, use the nucleus, but eventually get the protein into the mitochondria, which has always been one of the more challenging steps. We have a very strong basis in manufacturing this gene therapy, and as I noted, we have 5' and 3' components of this mitochondrial targeting sequence taken from the COX-10 gene. So the schematic here is to put the gene construct with the mitochondrial targeting sequence into a vector and do intravitreal injection. Again, a safe technique that's been employed more than 1 million times over the past decade. Once the therapy is in the vitreous, it can then get to the retinal ganglion cell. It enters the retinal ganglion cell through endocytosis. The gene is delivered to the nucleus transcribes the gene, the mRNA enters the cytosol, and because of the mitochondrial targeting sequence, the mRNA is associated with polysomes near the mitochondria, and the protein enters the mitochondria as it's translated, incorporates into complex 1, and hopefully restores mitochondrial function and prevents apoptotic cell death. We've hit a lot of milestones. There's more to go, but that's okay. We have a strong proof of concept in animal models. We validated this data in vitro with LHO and patient fibroblasts. We have a very good basis in preclinical non-human primate toxicology program, and now we're proving that excellent safety with our phase 1/2A study in Paris, a first-in-man trial that is a dose escalation trial. And I'll let you read the title, it's a little bit long, but it's going very well. I won't get into all the details. Suffice to say that we're very pleased with the safety and tolerability profile, and it's going to let us go on to phase 3 in the very near future. Suffice to say that there were no unexpected adverse events in patients, and there were no serious adverse events related to GS10 in our patients. So we're dedicated to getting this quickly because we know that there's a very big need. We've been in front of the FDA and the EMA a number of times, and that interaction has been very successful and very helpful. We've submitted to the Recombinant Advisory Board of the NIH, which is a requirement if you're going to perform gene therapy trials in the US. And we plan on submitting our IND and CTA in the next few months, and then initiating the phase 3 trials in the beginning of 2016, and hopefully getting the data we need and the proof we need to go forward and have an effective therapy for patients. The trial is going to be held in a number of sites throughout the EU and the US. In fact, I would ask you if you have any interest or you know of any patients that would qualify to contact Professor Corelli's team, Piero Barboni, and specifically I would recommend you contact Chiara Lamorga and Michela Carbonelli, who will certainly be willing to help you. If you know people in France, this will be done in Paris at the Cannes-Vain Hospital, the Centre Hospital Ophthalmologie de Cannes-Vain. In the UK, you have Patrick U.I. Mann at Newcastle and at Moorfields. In Munich, you have Thomas Klopstock. And in the US, we have 3 really great sites. Alfredo Sadoun, who you've heard about, at Doheny, Nancy Newman at Emory, and Mark Moster at Wills Eye Institute. I think it will be important to explain to patients the risks and the benefits of undergoing this, which of course all of our investigators will be armed with, and also the fact that certain patients will qualify and certain patients will not. Our gene therapy only targets the ND4 11778 mutation, but the hope is that once the platform is proved, we can then go forward and find the ND1 and the ND6, and then again apply it further to other mitochondrial diseases, other neurodegenerative diseases, etc. So in summary, I think we're all here for one reason, to find therapies for patients who don't have it. The eye is a good place to start for a number of reasons that you're all aware of. Our goal is to use gene therapy with AAV-based technologies as part of our platform. And I think that a number of these components of the platform has already been proven. So with that, I want to say thank you very much, and I appreciate your time.